Good morning, everybody. I'm really excited to be here. I'm really enjoying this leadership series. I think I'm getting as much out of this as hopefully you guys are. It really goes to show you can learn about leadership your whole career and the tricks of the trade from our colleagues. So I also want to welcome our UK students. So if you guys can see us, welcome this morning. We're happy to have you here as well. Everybody say hi to them.
Hi.
Wish you all could be here. It's beautiful spring in Washington. You're missing cherry blossoms and daffodils and tulips are about to bloom. It's beautiful weather. So we have a really terrific program today. We have a terrific plenary speaker, a really great panel discussion lined up. We're going to have a little roundtable discussion afterwards focusing on precision medicine and what that means to us and how we can better understand it. I have to admit I haven't paid much attention to precision medicine, and now doing a little digging in about what it means and what it means for the future of medicine is really exciting, so it'd be fun for all of us to be thinking about that. So I want to just get started by introducing Marshall Fordyce. Is Marshall? Oh, there he is. Works at Gilead in California, but also joined the board of the Mary Lasker Foundation in 2012, and he's here to introduce our speaker this morning. So Marshall.
Great. Thank you, Tom. Welcome, scholars. And I think the whole purpose of this meeting is the world needs you to recognize and develop your leadership potential as scientists. And it's my great pleasure to introduce this morning's keynote speaker, Dr. Craig Thompson. He's president and CEO of Memorial Sloan Kettering Cancer Center in New York. And I think our best leaders continually build skills. They leverage resources, and they fearlessly create synergies. And Craig Thompson does just that. Dr. Thompson began his career pursuing a high-quality medical and subspecialty clinical education, undergraduate at Dartmouth, MD from Penn, internal medicine at the Peter Bent Brigham Hospital at Harvard, and subspecialty training in oncology at the Fred Hutchinson Center Cancer Research Institute at the University of Washington. He broadened and deepened his research experiences at the National Naval Medical Center, University of Michigan, University of Chicago, and as a Howard Hughes investigator. He then served for 11 years at the University of Pennsylvania as professor of medicine, was named their first chair of the Department of Cancer Biology, and became director of the Leonard and Madeline Abramson Family Cancer Research Institute. And then since 2010, Dr. Thompson has served as president and CEO of Memorial. His demonstrated leadership skills have been increasingly sought and for great purpose. He serves as the medical advisor, serves on the medical advisory board of Howard Hughes, the board of directors of the American Association for Cancer Research, a member of the Lasker Prize jury, and is associate editor of Cell, Immunity, and Cancer Cell. Dr. Thompson has been deeply engaged in translating his insights in cancer biology and immunology into developing novel therapies. As scientific co-founder of 3 separate companies, including Agios, which is currently developing novel small molecules against mutant IDH in multiple cancers, and it's in the clinic right now. What a fantastic career, and what can we learn from his path? So with this in mind, I'm excited to hear Dr. Thompson deliver today's Lasker Lessons in Leadership keynote talk, a discussion of the difference between management and leadership. So, Craig.
Well, I want to thank Marshall for that really kind introduction. I don't often actually agree to give lectures on leadership because I'm not really sure how to tell anybody anything. I think the most useful thing I can do is probably the next 3 minutes is to correct Marshall. That sounded really good. My mother would have been really proud to hear about all those things. The truth is I went to college to play sports. I didn't really know what I wanted to do. Started out as a government major because my dad was a military officer and I was a career military brat until that point. Sort of realized halfway through college I actually probably needed a job at the end of all this. Turns out the sports I played and was good at weren't ones you could make any money at. And realized the only things I liked were science courses. And so I turned to what happens to the average American, actually the average person worldwide. If you ask them, do you know a living scientist? What do you think their answer is? Who do they answer? Number one's Einstein. And then you remind them that he's not living and you don't know him. Which always throws Americans for a loop, okay? And so you say, okay, but now, do you know a living scientist? Who do they answer next? After you remind them that Einstein is not living and they don't know him.
Hawking?
What?
Stephen Hawking?
No, they don't answer. You come from a slice where that might actually be a viable answer, but for the public at large, it's not. The number one answer, over 60% of people, is their family doctor. And that's really where Americans get and think about science and where it impacts. They would like medicine to be a science. And so that's really what we're talking about. So I put together some slides. I don't know how we'll get through them. Mostly I'm going to tell you a little, again, I'm going to continue to correct Marshall and sort of now try and create a little bit of a bond with you guys. Once I realized that I wanted to go into the sciences, I didn't really know anybody that had a job other than the base doctor. So I applied to medical school. I didn't have any way to pay for it. My family didn't have the resources to pay me through graduate school. I was supposed to go get a job. And so I signed up for a military scholarship because this was right at the end of the Vietnam War and they didn't have a way to maintain doctors for the military. So the Navy agreed to pay my way through medical school if I could get in. Got into one medical school. It was the same place I went as an undergraduate, Dartmouth College. And 6 months through my first year in medical school, they said, we're changing, and you don't have a place after the second year. The reason for that is a 150-year history combination between Harvard and Dartmouth. Dartmouth had, for 150 years, trained in the first two basic science years, and then you transferred to Boston to get your clinical training. And they had decided to build in the Upper Valley of New Hampshire a new hospital, and Dartmouth students were going to stay there. And half my class had elected to do that, and half of our class was supposed to transfer. And in one day, I didn't have a way to finish medical school. So the reason I'm telling you that is because about half of you will bond with what I said next. You don't realize it, but I was in the first medical school class at the University of Cambridge. And how many people are Cambridge students here or spent time in Cambridge? So it's about half of you. You didn't know that your medical school only started in 1975, right? Same problem as Dartmouth. Cambridge's actually been graduating medical students for about 3 centuries. But nobody got an MD degree, or the English equivalent in letters, because everybody went and trained at the university hospitals in London and got their degree from the University in London, where they spent their last 2 clinical years in medical school. And so nobody got a degree until they built the regional East Anglia Hospital of Addenbrooke's Hospital. Those are the Cambridge people know where that is. Big hospital, big fancy place. I was there the first day it opened. There were actually 8 students that were accepted into the first class that were actually going to get an MD from the University of Cambridge. But this is England, and this is one of the great universities of the world, so they weren't going to do this with English citizens. So the first class, I was the 9th, I talked my way in, was in fact 2 Americans, 2 Canadians, and 5 dentists in town who all wanted to become oral surgeons and had thriving dental practices. And we were the first class at the University of Cambridge. And we had a full run of the hospital for over the next period of time. Now to get it, just so Marshall doesn't look bad, I did come back for a year and get my final training at the University of Pennsylvania to get a U.S. degree so I could go on and train. But it was amazing to have that experience across the pond, which is what you guys are all doing. So it's a real honor to actually be here and participate in what's actually an organized program that expects that exchange, because it really influenced me tremendously, both in science. I got a research job at the MRC building for those in Cambridge, and I actually lived at the hospital in the house staff quarters, because with only 9 of us, they gave us rooms in the hospital. The other reason it's sort of odd for you guys to invite me is that I don't have a degree like you guys do. I don't have a professional scientific degree. I have a trade degree. I'm an MD. And that's really important, I think, for you guys to think about as you go ahead scientifically over your careers. Nothing you know about science today will be relevant to its practice 40 years from now. And so you have to constantly learn the latest things, the latest discovery, the latest techniques, the latest new ideas about how biology is. We still know only a fraction of what we need to know about biology. And so one of the things about a course like this is to look around here. You're in a group now, 90 of you right now in the program right now, who have a common vision that you'd like to actually see medicine turn into science. That's why you come to an NIH and actually try and practice. And I will tell you, probably the only useful thing I'll tell you is that this is probably the most exciting time we've had up to now in human history on this. In my generation, scientists had to work on model organisms. We had to reduce the problem to simplicity. All the great genetic experiments started in yeast, then carried out in very simple invertebrate model systems. We were lucky we got to mice by the end, but that really told us some of the things that were possible, not necessarily how it actually works in the wild in a real outbred population, because nobody studies outbred mice. Anybody here work on an outbred mouse model? The sequencing of the genome in 2000, for the first time, gave us the power to do meaningful studies in the largest, best characterized outbred species in the world, and that's a human being. Human beings are really special as a species. We collect our broken into these things called hospitals, and we study and characterize them phenomenally, and we collectively put resources towards trying to figure out whether they can correct that. The next 20 to 25 years will be the only meaningful model organism, if we're really going to understand the next principles of biology, is going to be human beings and maybe complex plants, but you chose not to go into botany. So we won't cover botany today, okay? So that's really the preamble to this. There's two things about a leadership lectures that I want to take, and then I will take you through some things just to sort of hear about this. I never intended to be running a premier academic hospital and research institute like Memorial Sloan Kettering. Never crossed my mind. First thing I had to get over was I going to be a base doctor or actually be an academic? I actually spent 8 years in the Navy. I actually wore a uniform for 4 years across the street here. Ran a research lab, worked when they started a medical school as a faculty, and then realized I really liked the academic practice of medicine before anybody ever thought that I would actually do anything that had to do with leadership. I was lucky enough over time, not because I had any leadership training, but because, in fact, I just wanted to see the kinds of results we were getting in the laboratory tested and implied in a bigger and bigger group setting. And you have the opportunity as leadership to actually direct some resources to testing new ideas that you believe in. And so it's willing to trade off some of those responsibilities to do other things. I never, ever participated in a student government group. And so many of you will say, well, I need some leadership training, because all of you are in this room because you see yourselves as the emerging leader. And we actually, the people that picked you, that support you, that mentor you, actually see you in exactly the same light. So one lesson I'm going to tell you is don't worry about leadership. Right now, that's not what's important. You need to know our discipline. Whether it's getting a PhD and you're going to be a hardcore basic scientist or a translational scientist, or you're going to combine and be an MD, PhD, you need to be a good practitioner of that art. You can worry about leadership later on. The biggest waste of time right now, I'm saying something controversial, not every one of the faculty will agree with me. The biggest waste of the time that you will do right now is to get involved in some organization around being a student, then a postdoc. For the people that go on to medical training and are residents and fellows, the worst waste of a useful career is to be a chief resident. And the reason for that is they're going to teach you how to manage, which is what this other title was. They'll teach you how to schedule things. They'll teach you how to get everybody in a room together and get it all on a calendar. You don't need to learn that. Right now, you need to learn your discipline. You need to get deep into understanding whatever your lab's working on. Develop your own ideas about what you want to study and you want to concentrate on. And then don't be naive enough to think you can do that in exclusion of understanding everything else that's going on in biology. Because we sometimes get a little too much with blinders on. So be a generalist in that sense. So that's my preamble. That's probably the only useful thing I'll say. I think we're going to do this till 8:40. 8:40, right? So at 8:40, wherever we're all, we'll stop and take questions because that'll be more interesting. And if you've got a burning question, it will be more interesting to ask the question than anything I'm going to say, okay? So just shout it out. So I have some slides here. That preamble was to simply say that leadership isn't about management, and leadership is an interesting problem for medicine. And I'll actually have some slides about that and talk to you about a little bit about what I've learned about leadership in going from, first, can I run a lab with 2 technicians when I left my postdoc, to can I actually run a place? Memorial Sloan Kettering, we have 14,600 employees. We have 400 people that run laboratories. We have 1,000 physicians that come to work and take care of 137,000 patients at any one time. We see 600,000 patients in the clinic. We do 137,000 radiation treatments every year, 22,000 operations. It's fairly complex. How do you go from running a research lab with 2 people trying to figure out whether platelets initiate clotting, which is the very first thing I did in the laboratory, to that is impossible to understand even for myself. And I'm not sure it's what everybody should aspire to, but I'll at least tell you a little bit about what I've learned from that journey. So, with that as a preamble, what do you actually need to lead? The only thing you need to know right now is that 90% of leadership is common sense. And the biggest mistake really smart people do, like the PhDs and MD PhD students in this room, is convince themselves that they don't have to ask, does this actually make sense? Does my life experience say this is? And engage in what I'll say several times is magical thinking. And sometimes medicine is particularly good at magical thinking. Imagining they can reinvent human beings over again, or they can make biology what they would have done if they designed it. The most common thing my students have ever heard from me over the 40 years I've run a laboratory is, the one thing I'm really happy about is that I didn't design life. Because if I had, we wouldn't still yet be protoplasm. Because it's impossibly complex, biology. We still have only a fraction of the information that we need to know about. And you can't imagine the things that are going to occur new that you can't imagine right now. This is going to make me both look really old, but also actually put some things in perspective. When I was a first-year medical student at Dartmouth, there used to only be one meaningful scientific meeting where all physicians and all scientists came together. They used to call it the Federation meetings. Federations of American Society of Experimental Biology. All the older faculty are shaking their head yes. John, you have to say yes because we were on faculty together at the University of Michigan. And everybody, and only the really good academics were chosen to go because somebody had to man the ship. Somebody had to take care of the hospital. Somebody had to be in the classes. And so, of course, I wasn't chosen because I wasn't one of the academics. And so everybody that went that year in 1973 came back extremely excited, but it was extremely controversial. 2 things have been reported at the Federation meetings. They're always in Atlantic City, just to give some perspective. And there were 2 things reported that year. The first was something that everybody thought was a really interesting scientific curiosity. Some investigators at Stanford reported that they had found an enzyme in a bacteria that could cleave DNA in a sequence-specific fashion. Couldn't imagine why they had it, but they called that enzyme EcoR1. And the senior faculty finally agreed with the junior faculty we should at least tell them because that's interesting. It's part of the biologic realm of possibility. Today, you cannot do the practice of medicine and not know what restriction, Frank, length, polymorphism, and everything that's come after that teaches us about. But they had no concept that we would ever need to know it as a doctor, and they weren't going to teach it to any of the basic scientists. Today, everything you do is defined by molecular biology. So somebody was asking me at breakfast, how do I do all these different things? We didn't have molecular biology when I started. In fact, everybody that was run a really good lab dedicated to medical problems was a physician. So that was the training. That's not true anymore. Today, the intense training you need to just be a good basic scientist and the intense training you need to be a really good doctor are exclusive of doing anything else in your life if you want. So for the MD PhDs, you're going to have to pick. I think it's really great you're going through both training because it'll inform you, and I think humans are the best species, but you absolutely have to pick. If you want to be excellent at what you do, and that's why you're in this room, in the end, you're going to be a scientist who knows medicine, or you're going to be a doctor who knows science, but you're not going to be both and be world-class at it. And that's really changed because that wasn't the way I was brought up. So we were brought up in a slightly different time, and we learned it along the way. The second thing that I really want you to take away from these leadership lessons, at least to know my perspective, and the great thing about this is you're hearing from, there's no one leadership style. You're hearing from a lot of different people over the course of this. Take what works for you, and then just understand that there are others that have different perspectives. But ultimately, you've got to make your own. The one thing that's been true from when I had a laboratory with 2 technicians till now is always remembering the one thing that drives me crazy when I work in leadership meetings is everybody says, that's not my job. I got somebody else. That's the thing I don't do. If you want to be a leader, everything is your job. Because it has to get done. You're in charge of the whole thing, not, I got to do this. You don't get to be the CEO so you have the nice office that sits in the corner. In fact, you've got to prove to everyone that works for you, you'll roll up your sleeves and do everything that does to take and get the job done. And so the one thing a leader doesn't have is a job description. And the one thing I know I'm in trouble when I hire somebody new to our executive team is if they tell me what's their job description, I know I'm in trouble. Because they've got to understand they have to own the whole part of the organization that they're responsible for. And that doesn't matter. Your 2 technicians have to know that. They have to know that you'll got their back whenever there's something you don't do, they can do. And you've got to use that common sense to understand your job as a leader, whether it's going to be in your starting lab or anywhere further in your lab, to really understand what affects individuals that you're responsible for and what affects the whole team. So to give an example, the NIH is the worst about one thing that was the most valuable lesson for me in starting as a basic scientist. What's the one thing in your laboratories that can screw up everybody in the laboratory? It's all the stock solutions. And here at the NIH, you get to order them from an order sheet. And they get delivered. And they're made up somewhere else. You don't even know how they're made up. I actually think that's the worst thing a scientist ever gets trained to do. For the first 10 years I ran my own laboratory, even by the time we got to be 14 people in the lab, every stock solution in the lab for the first 10 years was made by my hands, by a formula that everybody got to see that was in a cookbook at the front of our laboratory. Because those things, those stupid things, the PBS and the media, they're the things that screw up everybody in the lab. More labs go down in the starting parts of their career because they miss the simple. They want to do the cool experiment with the cool antibody and the cool way of fractionating proteins. The stock solutions are what kill you. It's the common things that kill you. And leaders that pay attention to the things that can kill everybody that they're responsible for are the ones that get back. And that matters as you go through your research career. The second thing is it's really true as you start from a laboratory perspective. It's equally true when you start and become a resident on the clinical wards, is that if you're going to be a leader of a laboratory or a leader of a medical team, you actually want to strive to know to be everything, every job that's part of it, as I said earlier. But the one thing, as you get more and more people that report to you, what you really want to be in your laboratory is the second best at everything. Because if you don't appreciate that there's a new postdoc who knows how to do this technique and is really the best, and you don't learn from them, you'll never get better. If you don't understand that everyone on a medical team is good at some aspects of medical procedures and not good at others, and understand who the best is, you won't get the best out of your team. And if you don't respect the person that is the best at something that works for you and follow their lead, you'll never get anywhere. On the other hand, they've got to respect that you understand how hard it is to be the best on your team at something. And that's why you've got to strive to learn how to be second best. Learn from them how to actually do it. One of the things I do in my laboratory is every time we get somebody new, we ask them to teach us everything they know how to do that we don't do. And we make them learn a couple of things they don't know how to do, even though they might not be relevant to their project. I do that only to figure out how they learn. And the technicians always tell me, and it's been a perfect test over the last 40 years. I send a postdoc and say, go learn this technique that the technician's really the best in our lab at. If I come back 6 hours later and they're not still together, I know that postdoc's never going to make it in science. Because you can't learn everything by learning the SOP that the technician. You've got to follow them through. You've got to ask, why did you add it that way? How do you do that? Why did you wait 50 minutes? Why can you stop here and not there? All the things that aren't written down. We still are an apprenticeship trade, and you have to be able to know how to do that. And it doesn't matter. Medicine, science, that is the number one skill. And so learning how to do that and what matters in everything you do gives you the ability to help others and to instruct others as you go forward. That's sort of what I think. Second thing I wanted to actually say is there's some unique things about medical leadership. I was actually at a retirement party on Friday, and it was very nice. And it's an important leader for us at Memorial. And they were talking about how he transitioned to leadership. It was actually his family. And they said he'd gone out and read this book when he became a department chair, his family said. It was 10 days to get an MBA or something. You know, you've seen those books, because he wanted to learn about leadership. It doesn't actually work in medicine. We are the flattest organizations on earth. Every business school tells us we'll fail. We are the worst business model on the face of the planet, both scientific research and medical delivery, because we are absolutely flat in our structure. How do I know that? I know this probably better than anybody around. I have 14,000 people that come to work at Memorial Sloan Kettering. We only take care of one disease, only research one disease fundamentally. It's cancer. Every day, the vast majority of those people come in thinking one of 1,200 people are in charge. They're the people that are in charge of an individual laboratory. They're the people that are in charge of an individual medical team. They're a nurse leader on one of our floors for care. And everyone that works on that unit thinks that leader runs Memorial Sloan Kettering. The great thing that separates us from a lot of other places is the leaders believe it too. So I come to work every day with actually 1,200 people knowing they're in charge of Memorial Sloan Kettering. And the great thing about Memorial is that we end every day with 900 of them still thinking they're in charge. And those are the 900 units that really had a great day, really got some great research done, really took care of our patients well, really engaged in meaningful clinical research. There's 300 people that, of course, the day figure out they don't have something that they need to actually deliver what they're supposed to be doing as a leader. And somehow, by the end of the day, they figured out they can blame me, because I'm actually got this title of President and CEO. So you got to be willing to have 300 people that actually think they run the organization yell at you every day if you want to be a leader. And the only good thing about my job is it's a different 300 people every day. So 1,200 people show up in the course of a month into my office, but that really is how it works. We're incredibly flat. The perfect day, it's not happened in the 6 years I've run Memorial Sloan Kettering. The perfect day would be if all 1,200 came in and left thinking they were still in charge, and all the people that work with them thought they were in charge. Just doesn't work that way in this big an organization. So you're there to back them up and to make sure the next day they begin and end in charge. That's actually what leadership's all about. Your postdocs can do great, your technicians can do great if they've got all the reagents and you've trained them right. And so you don't really, again, the perfect day hasn't happened, would be if I didn't have anything to do. We are incredibly horizontal. So if you think about it in science, the NIH here, where you're doing it right now, is a really great place to look at it. Think of how much different types of medical expertise go on at this institution. From people who are world-class in microscopy to crystallization to cell biology to actual clinical and translational research in the hospital with real patients with extraordinary rare disease. It all goes on at the NIH. There's not any person that could have all of the expertise necessary to do all those things. So as opposed to most business school kinds of education of organizational and leadership structure, they tell you have a hierarchy, everybody has 8 people that report to them. It doesn't work that way. What medicine and science in your careers is going to be about is about building collaborations with enough expertise to attack any scientific or medical problem with enough range of expertise to be sure that you're not fooling yourself into just finding an association, but actually getting at root cause kinds of issues. And so look around you. Your best friends out of this program, out of the people that have trained themselves in different parts of the discipline than you, and you ought to stay in touch with them for the rest of their lives because you know what their strengths are and what their expertise is. The people I was an intern with are still the people I call for medical advice because I know exactly where they go and I know what they were good at and what they were bad at. It's the same as true in the laboratory. When I want to know about how you think about DNA, I talk to one set of people I trained with over my postdoctoral time. When I want to know about gene expression, I talk to a whole different group of people. The number one thing you guys have to change, and please do it before I get some really bad illness, is that it remains true and has been true throughout my entire career. It's actually why I switched from being a clinician into being a basic scientist, is that medicine is not yet a science. And so many of you that are trying to be MD, PhDs are going to be really fundamentally disappointed. You're going to get on the wards, and you're going to find out that 90% of what we do in medicine has nothing to do with science. And there's no level 1 or level 2 kinds of evidence that this actually has a scientific basis. On the other hand, the only hope is in the next 50 years, your generation and the people you train will turn medicine into a science. And science is the true future. And now, as I just said, I actually think the sequencing of the genome was the transition. You're going to hear me say over the course of the day that I'm not as excited about molecular kinds of things, but absolutely it underpins everything we can do to study complex biologic disease and how complex metazoan organisms answer that question. It's going to happen through the scientific study as part of medicine. So I think that's actually everything I want to say. I was going to take you through, I've got 5 minutes, a demonstration project about how you lead. And I think this matters. We probably won't get through it. That's okay. I really thought of 6 things that really you have to remind yourself all the time as a leader. And it was equally true when I got my first technician and I ran a small laboratory in the New England nuclear building. My first research job, just so you know, remember I was in the Navy. So when I finished, and again, you got to make your own opportunities. That's another thing I actually do want you to say, and I think you guys understand that because you're actually in a program you couldn't possibly have started with. So you started at different schools and you found this as an opportunity, this wonderful program that you're in. I actually think that actually defines people that are on their path to be translational, really fundamental changers of fields, are people that will find new opportunities. And so when I was finishing medical school, after this saga of going to 3 medical schools, I realized that I actually had to go into the military. And at this point, I actually was really interested, because of my experiences at Cambridge and at University of Pennsylvania, in actually academic medicine. And my job was that I had to apply to every military hospital for an internship. So I took every penny I had ever had and saved, and I flew to every Navy hospital in the country. There were 6 of them at the time that had internships. They were all over the country. And I met with the head of the hospital. This is incredibly audacious. You should not do things like this. And I said, you don't want me. You'll be much better served if I go off and do an academic internship and residency and then come and spend 4 years in the Navy. And I promise I'll come back if you want me then. And I sweated it out. They'd never had anybody say this to them. And I got a letter in the first week in February that said that they were disappointed I was not one of the 392 best interns who had applied to the Navy for an internship. And I would have to find my way to another civilian internship. And the following week, I got a letter from the Brigham saying I was going to be one of the 20 House officers at the Brigham that year. That was a transformation to me. For the first time, I met people who really believed in academic science. You're in a class exactly like that. You guys all believe in this. You all believe that this is the place where medicine and science are going to come together, and that's why you're in this program. So the first time I met them, because back then it was an internship group, half of my group mates were MD, PhDs. I didn't even know they existed. And yet half of them had written papers in Science and Cell, and I was just lucky I knew a little bit of medicine. And I did a little more research, and I discovered I wanted to be like them, and the Navy didn't have that option. So what I ended up doing was, with Harvard's permission, I dropped out of my last year of residency, because it turns out the government doesn't know how to deal with failure. And if you don't complete your residency, the only thing they can do is put you into the research part of the Navy Medical Corps. So I was assigned, because I was a failure, I was assigned to work at a place called the Naval Blood Research Lab. If I'm ever doing something useful in life, I actually think that job was probably the most useful thing if anybody ever writes about me after I'm dead. The most useful job. My job in the Navy for the next 2.5 years was to take care of 37 freezers. A couple other things that I won't tell you about, but 37 freezers. Those 37 freezers were in the basement of the old New England Nuclear Research Building. Back when they were selling restriction enzymes, they'd sold it to the Navy to keep these freezers and to keep the 5 upper floors to do research on blood components. The Naval Medical Research, or the Naval Blood Research Laboratory, had been founded after World War II, because World War II was the first war on Earth where more people died of projectile induced bleeding, that means a bullet shot you and burst an artery and you bled to death, than gangrene. And so they needed to figure out how to resuscitate soldiers in the field. And that meant they had to be able to deliver transfusions in the field anywhere in the world. So there were 2 things they were tasked with. Can we freeze blood? And can we unthaw it quick enough to resuscitate people so that you could have a bank of 10,000 units of blood? The Navy was assigned this because the Navy had ships that could go throughout the world. Unthaw it quickly enough that it could actually resuscitate a soldier in the field to get them back to a field hospital to rescue their lives. And over the next 10 years, that's what these guys did. And the Korean War came around just at exactly the right time. It was a war just like World War II, and that most people died of the bleeding from the projectile injuries. And you've all seen the MASH unit movies and all that kind of stuff. That was all real. And the unit that I worked in now 30 years later had figured out ways to keep blood products to last longer. First, the way they work in your blood bank right now. Up until then, you could only keep blood for 3 days. They worked out first that you could do it for 42 days. That's what every blood bank now uses in the country. They figured out how to freeze bloods. Every single person works at a cell biology lab, and you take out a vial of frozen bloods and you unthaw it. That was reduced to technology in that laboratory. No one knew how to freeze living cells before that day. Used throughout the world. They're the guys that figured it out over the next 15 years. They then tried hundreds of different solutions, and those 37 freezers were all collected from Korean war vets with different preservation solutions. And my job was once a month to find one of those vets, bring them back in, completely type and cross them, run a little mini blood bank, and then give them back their unit that had been stored for 30 years. And simply because we chromium labeled them, ask how well did they circulate, test whether they could still carry oxygen, and whether they could actually work as a resuscitation vehicle. And we reduced that to practice. My research, they give you a little bit of time. That was my actual job. I had a little research because they wanted to do it with platelets. We still can't freeze platelets, but we worked out all the technology that now allows you to keep platelets alive for 5 days and separate them from whole blood. That was not done when I was an intern resident. That story's only interesting because I worked on something I actually cared about. I really liked hematology from my residency. To get a platelet count when I was an intern, you had to order a hematology consult, and the fellow had to come and draw the tube of blood themselves and count them by hand. We worked out and reduced the machines that actually did the culture counting that you now take for granted in hospitals and that you use in laboratories to count your cells and your cell culture. All of that was reduced to practice in this unit I did. Today, you're on the U.S. highway system, you get in a bad accident, you have a rare blood type, the only reason you survive is because every blood bank in America has blood cells, rare blood cell types in a freezer. And the greatest thing we did in my 2 years was to figure out how in 30 seconds to unthaw using microwaves a unit of blood. Purely engineering, but every day somebody's life is saved because of that. Coolest thing I've ever done. Done lots of fun science, but don't underestimate the importance of the engineering that we're doing and reducing things as proof of principle down to practice. People thought it was idiocy to use a microwave to unthaw a unit of blood when we started. We blew up a bunch of units of blood, let me tell you. Okay? What I learned from that and from all the lab experiences I've had is just these 6 lessons. Maybe think about them, maybe not. You absolutely have to engage your staff and have a sense of mission. So today, that isn't the same as convincing my 2 technicians. We were working on platelets and they were the coolest things on earth because nobody knew what they were. Today, I've got 14,000 people that come to work, and I have to give them a sense of mission. And it's really not you guys that I have to convince. I actually have to convince the guys that park our cars, because in New York, parking is a real problem. I can't pay our parking attendants very much. They get paid a barely subsistence wage. They get keys thrown at them at 9 in the morning when our first patient clinic's open. We only have 250 parking spots. They get keys thrown at them from 2 blocks away. Say, we're going to be late for our cancer appointment. They've got to take that car off New York City streets, park it safely, keep it intact, and get it back at 5 o'clock when that family goes home. They have to believe, even though they're being screamed at, not because it's them, but because of how much acute anxiety there is. They've got to believe in our mission. How do you do that? It's interesting. The only people that have parking spaces there are myself, because I have to get around the city to represent us, and a few of our surgeons who have to be there all times of night for life-saving surgeries. The surgeons still haven't figured out how to connect with that staff and give them a converse. They come in and expect it to be parked. I learned early on to connect with those guys. I had to understand what they did. So for the first 2 months when I came in, I parked the car myself. And over the next 2 months, as they realized I'm not, have you ever seen a parking attendant in New York? They are world-class parkers. They can get within a quarter of an inch. Over the next 2 months, they taught me. They're incredibly proud. They know I'm not as good as them. They showed me how to readjust the rear view mirrors so that you actually saw the line so you could pop down a line perfectly. They are so proud of that, and that I try and park my car, that they actually buy that I understand what they're doing. So when I ask them to do something special, like when we had a 26-inch snowstorm, they'll go anywhere. You have to understand what their job is. They have to believe you know how hard and how little they get paid for it. And you have to do that throughout the whole organization. Your technicians, I'll bet if you go back and think about your own laboratories, the equivalent is your technicians. You have to engage them. They have to know. They're not just cleaning the glassware. They're not just doing this pipetting. One of the nicest things I thought about this last night. I was on my way here from the airport last night to the dinner, and I got an email, 2-page email. Who writes a 2-page email? From a young guy named David Wang. It's a really nice letter. I hadn't heard from David in 20 years. He had been a work study student in my laboratory at the University of Chicago. He was a nerdy little history major. He needed the money. He wasn't making a lot of money. We got him hooked. He actually stayed 1 year after college and became a junior technician in a lab, then said, no, I'm going to go on and do my life. He wrote me an email to say, just wanted to let you know, done pretty well. I now run a consultant company. I went and got an MBA. I run a consultancy about biotech startups in Boston. We've gotten pretty successful now. So I gave myself a year sabbatical because I don't have to work. I've got enough people to work. And I'm going back getting a biotechnology master's degree, so I really understand what they're doing. And the professor yesterday covered one of your papers, and I got to tell them about what it was like to be a junior tech in your lab. That, to me, means more than the fact all the people that are professors that have gone on as postdocs. If you can connect to everybody that works for you and get them motivated and get them interested in it, then you're going to run a world-class lab. So you have to engage your staff. You have to get them to understand what the mission is, whether it's that particular project in your lab, and you can't discriminate among the people that work for you. You've got to make them all believe, from the technicians to the best postdoc, when you have your own lab, that they're equal. The thing that separates us from everything else is that we have to know our business first. Today in a business school, they tell you any good leader who goes through a business school should be able to run any major corporation in America. It doesn't matter what widgets they sell. It does in our field. You have to really know science if you want to run a scientific lab. You really have to know medicine. This is not about management. This is about leadership. You cannot shortcut. I spent 9 years as a postdoc learning molecular biology. I don't have a PhD degree, but I spent more time in a lab than 90% of the people who say they have a PhD degree. There's no shortcut. I had 3 different postdocs before I was comfortable I could be a scientist. Medicine, in the end, is an apprenticeship. Right now, the best thing you can do for leadership is to apprentice yourself to somebody who you admire, your lab leader, the leader of a medical team on the floor, and learn what they do. Learn why they do it. Ask them why they did it. That doesn't mean you adopt it. You've got to find your own style of leadership. But you need multiple good mentors. And sometimes, actually, one of my mentors is actually, he doesn't like me, he's actually heard me say this. He's a great scientist. I learned a great deal about how to do science from him, so I'm not telling you who it is. But every time I get to a leadership problem, I ask, what would he do? And I do the opposite. And it works great. And I'm not sure, he's still an extraordinarily successful guy, has probably a lab of 60 people. But it's not my style. And so you have to figure out your own style over time. But you first have to know the business, whether it's medicine or it's surgery. And then I didn't have a chance, because I took way too long, to talk about this issue of magical thinking. We're going to talk about precision medicine today. We're going to have a big debate about it. 80% of precision medicine today is magical thinking. So I'll throw that out as the start. I'm not going to give you any examples. I had some built in here. We talked about it at breakfast, some of us here. But 20% of it is the future of science, and we've got to sort that out. And that's going to be really important. If you're going to lead, you're not supposed to be mom and dad. And that's a unit you got to get away from and understand. You have to know how to challenge your staff to get the best out of them. Some of them it's by yelling at them. Some of them it's by propping them up when they get disappointed, because 80% of scientific experiments fail. And we live in an oddity. We can't have 80% of our clinical care fail, but 80% of our research labs fail. And so there's a real dichotomy there, particularly for the MD PhDs who have to go back and forth. But in the end, you have to challenge people to be the best. And then don't obstruct them when they make a decision and they try to follow through. Give them the support they need to do it. It's the same thing as once you make a decision and give this to somebody else, don't go second guessing them and say, I wouldn't have done it that way. That's the worst way to undermine as a leader something. And then the most important thing, which is what I just said at the beginning, nothing you know now will be useful to whatever you're doing in medicine or science 40 years from now. You'll be appalled at how naive our notions were. And I just told you about a story from 1974 to now. It's going to be equally true all over again. In every 40 years, somebody could have told that story for the last 150 years. And so don't forget that as we go forward. Okay? I'm going to stop, take any questions. I didn't get to any of the real examples, but I told you a few stories, and I'm happy to talk at any point along the course. Thanks for having me.
Okay, so we have about 10 minutes for questions. So if people have questions, feel free to fire away. Don't be shy.
Yeah.
You said that medicine is not yet a science,
Yeah.
and you singled out the human genome as a big difference. Is there anything similar on the medicine practicing side that could happen to that transition?
I think the example I use when medical students ask me that is the following. I had the program. One thing great about starting at Dartmouth, where they only taught the basic scientists, is that they had some of the really great basic science chairmen. And so we had the guy who wrote the leading textbook has been for 100 years for pharmacology, Goodman and Gilman. And Dr. Goodman was our professor. He walked in the first day, the Goodman and Gilman book, which doesn't happen anymore, but it exists, but it's only in electronic form, was literally this thing, okay? And he basically said, as a doctor, they're going to tell you you need to know that. This is the leading pharmacologist, at least in academic practice in the country, and he says, but I just want to tell you, there's only 10 drugs here that matter. And if you know how those 10 drugs work, understand their pharmacodynamics, how that mechanism works, you'll be the best doctor of your generation. I'm not sure he was right, it was 10, it was probably 20. Okay? The problem is, we've gone by 40 years, and today it's over 1,000. So we do have 1,000 drugs that have a real mechanism and basis of action today, and no one can learn all you need to know to safely and effectively dispense those and the combinations and side effects. The biggest problem for medicine right now is we think that those are absolutely precise, that they only work by inhibiting that enzyme and nothing else. And we've gotten ourselves into some magical thinking that every drug can be perfectly safe. It will only go and find just this one enzyme and inhibit it and not touch anything else. And that's not the way biology works. And so we have to understand, it's the biggest problem. We've thrown away a number of really good mechanism-based disease from clinical medicine because they had a side effect. And the side effect was completely tolerable. And we forget that we are giving most medicines to people that are ill, and we're looking at a trade-off between the side effects versus the benefit that medicine gives. And the example, many of you have probably heard, certainly the ones in medical school, today, neither penicillin nor aspirin could get through the FDA based on clinical trials. You could not get them approved. And yet they're the two most useful medicines in the world. And so, again, we have to start to understand that medicines aren't precisely targeted things. It's a little bit of my problem with the magical thing of precision medicine, that it'll only go and find that. We don't know how to do that. And so we have to accept that biology is just a little more complex than that. Yeah?
I found your discussion interesting of how science is such a hierarchy.
Yeah.
And how it seems that your point is that that's a good thing because we need to be able to collaborate with people who have all
I don't have a point that it's a good thing.
Okay.
It's just that's the way it
Just
is.
how it is. But I think I've in some ways found it problematic in my PhD so far that someone in my room in London, I drove crazy because she was the only person I could ask for help on how to set up a field study.
Right.
But she wasn't my official mentor, so it was somewhat unclear to her how my taking up so much time of hers was going to be mutually beneficial. So how do you, as someone at the beginning of your career, how do you get people on board with actually mentoring you when you need them?
I think there's only one useful tome on that. I think it's Huckleberry Finn. I tell all students to read it. I tell them to learn what Tom Sawyer's did. He got the town to paint a fence. They had to bring the paint. They had to bring the labor. They had to provide the brushes. And he got them excited about it. And that's, actually, I think Mark Twain figured it out. That is probably one aspect of leadership you all need right now, which is how to coerce other people to engage in a project and not only not feel like you're taking their time, but being excited about it. And so one thing you have to do is you have to share credit. And that's a big problem right now about how we do this in publications. But we got to get over this idea I can only be a first or a last author. I think you have to be genuinely engaged. And so my argument about that you have to be the second best, over the course of my career, I'll just give you some ranges. I can't possibly be good at this. We did for 3 years at Ann Arbor a field study. We bred Muscovy ducks in a farm, in a barn on a farm to do pure genetic experiments. You don't even want to know what it is, but I had to engage a farmer and his wife who sold Muscovy ducks are the lean ducks that you find in fancy restaurants. And they did this after they'd retired and rented out their fields. They did this for a living to sell it to all the high-end restaurants in the Detroit area. And I had to convince them that it was exciting not to breed them to have more ducks, but to breed genetic pairings so that we could study the molecular biology of the immune system in ducks. You got to figure it out. My postdoc, who now runs his own company out in Baystock, went out 3 mornings a week and sat at their table and let her, it was her, I'm so basically sexist, cook him breakfast. And she could reminisce about the great farm and what it was. And this was something useful she could still do, having run one of the great farms of the Midwest. That's how he coerced her. We had to learn how apoptosis worked, okay? We knew that the enzymes that we work on, BCL2, form channels in membranes. We had no idea how to do black lipid barriers. I had to read every textbook there was. We drove to University of Loyola every day to a group that used one of these Faraday cages and black lipid membranes, made the proteins, and we had to listen. I sat there for 8, 9 hours, not really knowing anything we were doing, to get the channel recordings that first gave us the insight that BCL2 proteins poked holes in membranes the way bacterial toxins did. I then had to defend that in a national meeting with 5,000 people, and the first question up was, how do you think the Nernst equation works to regulate this? So I actually had to draw out the Nernst equation. Nobody ever trained me in the Nernst equation. I had to know enough. I had to learn it from these guys well enough that I didn't embarrass them so that they thought it was an equally important achievement. We crystallized, back when we didn't know what transcription factors like, we were lucky enough to collaborate with some people who are great X-ray crystallographers. We crystallized one of the first transcription factors that was crystallized as part of that collaboration. 3 months later, some scientists from Rockefeller University said we were idiots and we didn't know what we were doing. And look, Thompson, he doesn't know how to do X-ray crystallography, and said we were completely wrong. You get totally frightened out. I was working with the journal, Junior Crystallography. We went back through all our data. We had to learn more X-ray crystallography than anything else. We were sure we were right. A month later, they retracted everything they said because they had misassigned a backbone, an NOE. You had to know that. You had to actually be comfortable with that. You had to share in the discipline. That person that we did the X-ray crystallography now runs the largest group of crystallography in academics at Vanderbilt University. But we had to have that collaboration. We had to get over actually that quite painful time to be able to do that. And so, and you build up trust over time. So if you have 4 of those examples, people will join on for the 5th. And they'll be interested in that. And they'll teach you things. And it has to be about joint collaboration. You're on this not to extract things out of people, but to be on this road together, and both of you have to be equally successful.
Besides being a leader in the lab, so how did you make the transition from being a leader of just your lab to being drawn into a leader of a major institution? And we know you outside of New Orleans, Sloan Kettering, as a great scientist, many great publications, cutting-edge leader in science. Why did you make that decision to do a bigger job as a leader of that institution?
So first of all, no one, a couple of people do. I actually don't know how they do it. No one makes the decision just to go from running their laboratory or their medical unit to deciding to run something that has 14,000 people and more moving parts than anybody can keep track of. You convince yourself over a long period of time. So my transition was I ran a lab in the Howard Hughes Medical Institute, and I really liked what we did in the lab, but I actually thought we had more to teach immunology. We were the only lab in the world that did immunology at the time that didn't study mice. We studied chickens and ducks, as I just told you, and we studied human beings. And we'd found some pathways that we couldn't convince anybody were meaningful. You actually all know them now. We discovered first the CD28 pathway and then the inhibitory pathway, CTLA4. But because we'd done it in these species, nobody believed us because it hadn't been done in a mouse. And so I was given an opportunity to come and run a new immunology research unit at the University of Chicago. They said, we want somebody actually that would actually do human. And so I ran a place, my first job in academics was to populate a place called the Institute of Medicine at the University of Chicago. It was a perfect place for me because it was the research building attached to all of our undergraduate teaching. So for the next 5 years, I was where all the undergraduates came if they didn't like how their classes were going. So I learned all about that. And at the same time, I got to populate a 5-floor research building with 25 other laboratories that were doing that. I've mostly focused on the immunology part of that. We didn't screw it up bad enough that when somebody else wanted to start a research institute that was going to be slightly larger, they said, geez, you should consider coming because you did this on a shoestring. Now we'll give you 10 times more money. Start an institute here, in this case at the University of Pennsylvania. And I was dumb enough to say, geez, it's like everything in life. If I could just do it over again, I could do it a whole lot better. And suddenly you're doing something 5-fold larger again. And then you realize it would be nice to just concentrate on one thing. Part of the problem working in a large academic medical center, if you're only working on one area of it, and the big problems right now, the big real questions right now that occur in medical schools are what are we going to do about neuroscience and neurologic disease? What are we going to do about cardiovascular disease, and particularly diabetes as an end risk of cardiovascular, and then cancer. Those are the three big, and you're always fighting for the resources because the university has X number of resources, X number of faculty, and that's the biggest job you find when you're actually running one of these units. So for 20 years I joked, the only job I could imagine that would be better than the one I have would be Memorial Sloan Kettering because everybody knows where they could go. I never thought it would ever happen, and then one day the board showed up in my office and said, how'd you like to come? I never applied, and it just happened. I never intended, was a joke, I will admit that. Many people will tell you I used to joke about that. But you don't set out to do this. It just sort of happens. I'd be perfectly happy running my lab right now. Quite honestly, if you could, I would go to any of the labs you guys are working on if you could give me back 40 years of time so I could do this over again for 40 years. It would be incredibly cool to do again. There's nothing special about being the president. It's a little more complex. I've got more people to do it, but all the lessons I learned in my starting lab at the New England Nuclear Building and the Naval Medical Blood Research Laboratory are exactly the same I use today. And that's why pay attention to everybody in your lab. Pay attention to the lessons your lab leaders do. It'll serve you wherever you end up. But one thing I'll promise you, this is the coolest career you can think of. We're planning my 40th medical school anniversary right now, and not one of my classmates who've gone out to practice medicine, one of them chairs of medicine, distinguished thing, are as excited about what they're doing as what you get to do if you're engaged in the research vision. So the 3 or 4 of us that still do research are by far the happiest with our careers. So it's great to see you guys.
Let's thank Craig one more time. Okay, I do have one question from the UK, but while I ask this last question, I'm going to invite the panelists to come up and take a seat so we can get the next
Come on up, guys. I'm taking up too much time.
So this is from Nick Adder. So this is a virtual student from the UK asked,
right.
What sorts of leadership roles do you think we should look for, avoid as PhD students or postdocs? What should we look for and what should we avoid as students?
So I don't think you're going to get the same answer. I think it's a really good question. I probably didn't go enough. I actually think the only thing you should do as a PhD student or an MD PhD student is learn the trade. I think you should avoid all leadership positions. I don't think there's anything that will get you, and what you have to get is a solid foundation is understanding of the discipline. And unfortunately, that makes you different than almost every other field, because all they're going to teach you in those jobs are management skills. They're going to teach you how to be an organizer of a meeting. You're going to have to learn that, but there are lots of times to learn that. And you have to get well-grounded in the discipline first. So I really don't think that's the time to circ out your leadership. Now, the MD PhDs are going to get lucky. They're going to get thrust into running a team on a clinical ward. Ultimately, you'll get an opportunity when you're a postdoc to have a student or two, whether it's an undergraduate student. That's the time those are the leadership positions you should learn, and you should take whatever you've seen from your apprenticeships, the various people you've learned from, and apply them and see what works in forming that relationship with somebody. So I think it's the first time you have somebody work for yourself. The other thing I say is if you really want to think about this, where leadership comes from, there's one thing I make everybody in my institution do if they want to come talk to me about leadership, and I actually meet with our emerging nurses and our emerging physicians on a regular basis. Every quarter, a group of them come. In the New York Times, and I'm not advocating that the New York Times is a great newspaper or not, every Sunday on the second page of the business section, they have a section that's called the corner office. And the first question is, When did you first have a leadership experience? They're fascinating. They are nothing that any textbook tells you about. And if you read it for a year, you will learn more about leadership than any other place I can find. Just the answer to the very first question. They get people from all walks of life, all businesses, nonprofit, for-profit, and that's always the first question. Read their answer. That's more useful than anything else I can tell you.
Do we have Tina here? Oh, okay. All right, I'm going to introduce Leanna Falcone, who's going to come up and introduce our panel. Thank you, Leanna.
Okay, good morning, everyone. Once again, thank you to Dr. Thompson for a most inspiring talk. It's now be very exciting to hear other perspectives as we all exchange on this topic. And I welcome our wonderful group of panelists here. So we have, first at the far right, Dr. John Niederhuber. He's the Executive Vice President and CEO of Innova Translational Medicine Institute. He's an adjunct professor of oncology and surgery at Johns Hopkins University School of Medicine and a deputy director of the Johns Hopkins Clinical Research Network. He is formerly the director of the National Cancer Institute here at NIH. He also chaired the National Cancer Advisory Board and led two great initiatives, the Cancer Genome Atlas and the NCI Community Cancer Centers Program, to name just a few. Next, we have Dr. Marshall Fordyce. He was already introduced. He's the Senior Director of Clinical Research in HIV Therapeutics at Gilead Science. He's previously an instructor in clinical medicine at the Rockefeller University and a research fellow at the Aaron Diamond AIDS Research Center. His current work is focusing on the development of tenofovir alafenamide for the treatment of HIV infection and looking at TLR7 agonists to reduce the HIV reservoir. Next, we have Dr. Cristina Annunziata. She's the head of translational genomics section of the Women's Malignancy Branch here at the National Cancer Institute. She completed her medical oncology training at NCI, where she did a lot of work on the NF-kappa B signaling pathway, which she expanded to ovarian cancer. And she now directs clinical operations at the Women's Malignancy Branch. We've already been introduced to Dr. Thompson. And so just to get the discussion going, I will start with a question that was actually brought up by one of our scholars. So I'll raise this to the whole panel, see who wants to answer this first. But in what ways do you think genetics, big data, and computer science will impact healthcare? Does anyone want to take this first?
I will give a simple answer, and I think hugely. I think that we're entering a very disruptive time in healthcare, and it's going to be very difficult for us to deal with the disruptive technologies that will be introduced into healthcare and still maintain our roles as the touchy-feely caregivers, if you will. I think it's going to be much more challenging for this generation, the next generation of our trainees, than it was when Craig and I started our careers. And I think keeping that balance between being the healer and being able to utilize maximally this huge amount of information that's going to be available to us. And also the technologies that are going to transform medicine from being episodic care to being almost real-time care. And we can talk more about that maybe down the road. But I think there's going to be a huge amount of change at a much more rapid pace than we're used to.
Speaker? Yeah. Okay, so I would definitely agree with what Dr. Niederhuber said, and also bring in what Dr. Thompson was saying about the sequencing of the human genome. I mean, that was really made possible by having computer science and being able to handle large amounts of data. I think how it applies to medicine, as Dr. Niederhuber was saying, is that we have a lot of information about patients' responses to various therapies, but it's not all in one place. And I think that's probably the role of computer science and large data, to be able to assimilate all that information for the benefit of the patients.
Maybe I'd just add, from a drug development perspective, things are going to be radically different in 30 or 40 years in terms of how clinical, first of all, how drugs are targeted, how clinical trials are conducted and designed, how that eventually becomes a market, and how we have an important engine in this country and a few others of generating new therapies, and that engine needs to kind of get with the program in terms of how the genomics revolution impacts new therapies, and I think we're going to see it. It's going to be enormous. It already is.
Absolutely. Yes.
Just to redirect, Dr. Thompson. Thanks much for your talk. You touched on the concept of as to what you thought leadership was in terms of total ownership, and that's also something that Ralph Schneiderman touched upon when he spoke to us. And I just think that's a really interesting concept. And if you could further that with the help of the panel, I'd appreciate it.
Cancer, so I'll speak just specifically to cancer to give an example, is I think many of us, including the leadership in my own institution 10 years ago, thought we should plan. Charles Sawyer's at our institution, along with Brian Drucker, had just shown the incredible results with Gleevec. It was the first success of a targeted therapy. As I was telling people at breakfast, that original cohort of 15 people that had chronic phase CML that got Gleevec, Brian's still following them. Right now, they have a longer life expectancy as a group than their age-matched controls that were picked in 1999. So that's an amazing transformational drug. And it's a really important advance in our understanding of maybe the chronic pathogenesis, because chronic myelogenous leukemia is not something people die of. They die because it transforms into acute myelogenous leukemia, and this arrests that disease. So the bad side of what they learned is that all those patients still take Gleevec, or a next, second, and third generation of the drug that's a little more effective, because if they stop it, their disease comes back. And then they end up getting, unfortunately, a few people that have not been able to get back on to control go on and have a real natural history of disease. 3 years later, they die. So precision medicine hasn't actually cured anybody yet. And we went 10 years before we found the second. We went through 800 medicines to get to the ALK inhibitors, which I was going to show the example. One of the reasons women are still going up with lung cancer is the fourth leading cause of death in America right now is lung cancer in non-smokers. The amazing thing is a large percentage of them have ALK translocations or activation events, and ALK inhibitors are just as good as Gleevec, it's looking like, for that subgroup of lung cancer, but only that subgroup. And so it's a second real miracle about this, although again, if they stop it, it seems the disease comes back. So we've got more to learn about the disease. But the sad part, a mistake we made, we thought, well, if these kind of medicines are coming on, there's 800 in development, we don't need to worry about surgery anymore. We're not going to need to do surgery anymore. We are trying as a discipline, because if you watched read Sid Mukherjee's book, or you read Ken Burns' documentary, one of the big sad histories of the initial parts of cancer when it was handled by surgeons. Many at our institution over the last 100 years, we did bigger and bigger and bigger operations trying to get ahead of it. And now we know cancer is a systemic disease. And so we had this idea that we wouldn't build any surgical platforms. Well, we still take care of 137,000 people. Surgery hasn't been replaced 10 years later. We do more surgical operations every year to help patients with cancer. We do more minimalistic. We now do robotic surgery. We just opened, we had to play hurry up and catch up. Last year we had not a single open OR for any new capacity. We opened 12 new ORs that are entirely outpatient. They're all robotic therapy. We had to find people trained in robotic therapy for minimal disease. But we didn't own the whole operation. We got to believe magically that because Gleevec worked, the next 800 would work. And we didn't stay focused on the whole discipline of what we need to do. Don't get me wrong. I agree with Marshall. 40 years from now, it's going to be a real different game, but I can't change it right now. I still need somebody who does state-of-the-art radiation therapy. I had to raise $250 million to have proton therapy so we could safely give the therapy to kids. And we did that, as somebody asked earlier, in a collaboration with 4 other institutions, because no one could afford to do that, but have that as a testament. And we invested, even though we totally believed in Gleevec, we invested an equal amount of money in immunotherapy. And so we have 2 of the leading programs. There are many other good ones. In both checkpoint blockade, because that started with us and Jim Allison, who was with us, and secondarily CAR T cells. And if we had said we believe our own rhetoric about precision medicine, we had never have done therapies that didn't attack the cancer but augmented the immune response. So you have to stay focused on even if it's the research side, the whole mission. Not throw away the traditional stuff until you really know it can be replaced. Not believe in your own medicines. Don't get me wrong, we give Gleevec in 2nd and 3rd generations for the patients. It makes a difference. We have 800 clinical trials going on new phase 1 agents. We're not going to give up on that, but we're also going to pay attention to the entire spectrum. And we tend to, in science, believe our own rhetoric sometimes too. So that's really why I think Ralph was definitely, in his time at Duke, he was really good across the entire healthcare spectrum, keeping Duke on focus on everything. So I guess we really both believe that.
All right, any other questions? If not, I have one actually building upon one of Dr. Thompson's points, but to the whole panel. You talked about getting everyone on your team to believe in your mission, and that includes patients. So how can we get patients to believe in our research mission, both on a grand scale and on a day-to-day scale? And I'm just trying to recruit someone onto a protocol.
So I'll start with that one. So, I mean, I have to, just stepping back, a lot of patients are pretty much already engaged when they are coming to enter a clinical trial. So patients are very much aware of research that's going on, kind of self-select and are interested in the mission from the get-go. So I mean, I think it is not necessarily difficult to get the patients engaged into the research. A lot of patients that are interested will seek out individual investigators, individual institutions, and know a lot about it. I mean, I think the patients that might be difficult to get engaged are those that are perhaps not as knowledgeable of what's going on and maybe a little bit more hesitant to sort of enter and be a guinea pig. I think as the clinical trials evolve and we get more precision medicine and we get more trials that are very, very focused on particular diseases rather than broadly, then patients might be more interested in participating. One of the things that I think we need to go towards is, from a clinical trial standpoint, it used to be a 20% response rate was a good goal in a clinical trial. In a phase 2 clinical trial, if you had a 20% response rate, that was actually considered successful. I don't think we should have that in the future. I think we should be able to, have a goal of picking patients and selecting patients based on biomarkers or based on their disease. We know that more than 20% are going to respond, and that should not be our goal. I know that's probably much, much in the future, and we have a lot to learn before we get there. But I think that should be our goal. Not happy with just a 20% response rate, but really aiming for, we want to know who's going to respond, and we don't want to give the medicine to patients that we can predict are not going to respond.
Great. Maybe I'd just add that I think everything that Craig has said is relevant. You need to be, you need to consider everything your job, and you need to engage the people who you want to enlist in your project. Right now we're conducting HIV cure studies. There's very safe therapy for HIV, and the risk-benefit balance for a patient to take an immunotherapy to try to reduce their HIV reservoir takes a real risk. And so you need to be able to enlist those people. You need, if you're the researcher, you need to make sure you know the study coordinators and you visit them in their sites and you have a chance to meet them in their context. And you need to be able to believe the details yourself, know their challenges. It's a tremendous thing for us to be able to engage those patients. And maybe going on to selecting certain patient populations, I think we also need to make sure that we engage different demographics and take advantage of what Dr. Thompson referred to as sort of the most outbred population among humans. In HIV research, we've had mostly data on white men, and that is not what the HIV epidemic is how it's reflected right now. And so you need to take risks and change what's been done before and make sure that you engage all stakeholders. But yeah, it really comes down to the fundamentals that Dr. Thompson already addressed.
And I'll just make one point. I think part of this is what you just, I agree with everything that was just said. For what the public sees as fatal diseases and looking for opportunities, of which cancer probably leads the list but isn't the only one, the public is very concerned about being, at one end, a guinea pig. And so since they know we are doing well with about 2/3 of cancer patients, they're afraid not to have that chance. At the other end, they're afraid to go into a clinical trial where they don't get the medicine. And that has to be dispelled, as Marshall said, by really knowing everybody on the care team, everybody that's informing the patient. In cancer, everyone gets state-of-the-art, and then you get plus or minus therapy. The one change that is actually very controversial right now, but we have taken a stance as an institution, we more than 95% of the time will only adopt a therapy if at the time it reaches statistical significance that the therapy is having an effect, there's a crossover. So that every patient on the study at that point can get the medicine. And that gives them, they may get it 6 months later then or a year later, but they're still going to get the medicine and faster than they get it any other way. And that, when we have a team that can inform people right, really gets them through a lot of their fear. They're going to get state-of-the-art, and they're going to get this medicine if it works either first or second. And again, you have to, again, they'll still complain. New Yorkers are great at complaining. They should still get it because they know it'll work for them, but you can get through that. But the whole team needs to be aligned, and that really is why you have to pay attention to everything, because it's right from the orderlies that meet them in the trial unit. If they don't understand that's the common message and this is a trial and what the trial means, you get a lot of confusion. But it's exactly what you said. There's a large amount of the public that would, once fully informed, are happy to engage in clinical trials. And it is really the only way we're going to safely go forward with medicines going forward.
I'd just like to make one little addition to that. Having some experience now with the larger network out of Johns Hopkins, where we're working more on later phase trials, and not just cancer, across the board. There are many, many barriers to this process. We can't discuss those today, but one of the barriers is our healthcare system itself. Having lived most of my life in the protection of a research university environment, that's a whole different ballgame than taking it outside that environment into the community. And if you're going to do large trials, our phase 3 trials or some of our efficacy trials, things like that, the physicians do not have the resources in practice or the time to be partners, and they're much more of a barrier to getting these large studies done and getting patients on it. So it's a resource, really it's a resource issue. But there are other aspects of this that prohibit or perhaps decrease our ability to do the things we really need to do in large populations.
Other questions from the audience? Yes.
Emphasized a lot about apprenticeship in practicing science. but it seems to me that there's somewhat of a less emphasis towards careful apprenticeship nowadays, because it seems to me like I hear talks about, oh, you've done 3 postdocs, means you haven't done well because you couldn't get that faculty job right away, and that careful kind of work at the bench has been, if you take longer to master something, it might be seen as a failure almost. I was wondering your thoughts about that perspective and how to really be a good apprentice, but at the same time get over the idea that you have to be fast-tracked, that you have to be this successful all the time, leading the pack all the way.
I guess I'll start with this, but I think everybody else should chime in. I'm not sure you have to be the youngest assistant professor in the country. And I don't really buy that you have to be fast-tracked. I turned down, after my second postdoc, a full tenure track offer from UCSF to go and learn stuff in Seattle so that I could actually be what I thought would be a good enough molecular biologist. It doesn't matter if you're the first-year generation to run your own lab. It matters that you have the skills to run a lab for 40 years. And I think we are, unfortunately, our career is too long an apprenticeship system. It's too long to train in the professional discipline and then all the other things that we've talked about in leadership. I will tell you, at my 10th and 15th college reunion, it was the most depressing thing, because everybody else was talking about the house they bought and talking about the BMW they'd just been able, because they were now a partner in their law firm or they were now a vice president in their company, and I was still driving the department secretary's Volkswagen Beetle that I'd been able to buy for $600 because a tree had fallen and crushed its roof in, and they were going to write it off, and I offered her $100 more than the write-off value of the car. I drove it for 6 more years as a postdoc. But I had the best time of my life during those times. I got to go to work and ask questions, and I got to follow my own curiosity, and I had amazing colleagues. And now I'm the only one at my college reunion who likes their job. And all the rest of them are talking about retirement, and I'm just trying to figure out how to keep this thing going because it's so much fun. And so I really get the pressure, because there's all the other issues of life and family and significant others. But 40 years in, there's nothing more fun than a biomedical research career. And every day I just wake up, can't wait to get in and see what the lab and the institution are up to. And every part of my job I like. I wish I had a little more time to do it, but that's a whole different issue. So the important thing is to have the skill set to actually lead a lab. And that you can't shortcut. That's what I was saying.
So I definitely get it. I hear where you're coming from, which is that when you're applying for grants and you're in your 3rd postdoc, then your grant reviewer is a little bit suspicious as to why you're still in your 3rd postdoc. So I get it. I totally agree that you need to know your profession, and that's what Dr. Thompson has been saying all along. You have to know your profession. You have to feel comfortable with it. But you do need to show some form of progression, I guess, which is how I would say it, going from one postdoc to another. Why did you choose the postdoc? If you have a clear reason, you want to understand another aspect of it, you want to gain another skill, you want to be able to sort of incrementally have more people working with you, set up more collaborations. Those are all very good reasons. But I hear your dilemma, because when you're from the grant review standpoint, it's kind of a challenge. I think the advice would be, as you become more and more senior or more and more advanced in your postdocs, you should be having more of a leadership role within the project. Not just taking instruction from the lab chief, maybe instituting your own project, becoming senior author, corresponding author. Kind of go towards that way.
I think this is now getting very interesting. This is a great question, and I think you all confront sort of the careerism, if that's a word, and of what you're expected to do, what the next right step is, and I just think you got to listen to what Dr. Thompson said. You got to trust yourself, take your own time to build your skills, and if you don't think you're ready, you're not ready. You really have to trust yourself. So yeah, that's the most important thing in my mind. And I just wouldn't worry about careerism. You know, I'm the only guy working for a company. I drive a Prius with a big dent in it. And I think you have to fight careerism for yourself so that you clear out some quiet so that you can work on your skills. For me, and I'll challenge but not obstruct, I was a chief resident. If you have the opportunity to do it, you should. For me, it meant that I had a year to refine my clinical skills. I taught for 80% of that time. There was some management involved, but mostly it was teaching and tying research to the clinical cases that we were seeing at Bellevue Hospital with the greatest clinicians, and it was truly one of the greatest years of my life from a leadership and teaching perspective. Not to directly contradict, but we all have our own path. But really, what it did was clear out time so I could hone my skills, and that year made me a clinician that I will never lose that. So I like the focus of Dr. Thompson sort of saying, make sure that you trust yourself, because then when you're challenged at a meeting or you're challenged by a colleague or you challenge yourself, you know where you stand. And careerism it really threatens that confidence.
So I'll just give a follow-up to this. I completely understand what Marshall says. I will tell you that I've given that advice to every single one of my students, and more than half of my MD PhDs have ended up being chief residents. So, including my most recent graduate, who is next year going to be the chief resident at Mass General, and I spent my entire time trying to convince him out. He actually came down 3 weeks ago to New York to tell me he was not going to take my advice. And actually, that's why I know it's the right decision for him. I have what you need to listen from all your, and then make it, which is exactly what Marshall said, make it yours, okay? It doesn't matter what I think, it's what matters to you.
I want to just compliment Craig, because I really enjoyed listening to you today. And I've got lots of notes that I put down for my next career. It's going to help me. I'm looking forward to it. One comment on one of Craig's slides was common sense. And I think that applies to your personal life and your personal decisions as well. You've got to make those decisions with common sense. Am I going to grow? Am I going to be more at the end of whatever I take on? So when I decided a very difficult decision to go to a community environment, which I had never been in before, I said, well, that's exactly the reason to go. I've never been in this kind of a healthcare environment. I don't know much about it. In fact, I told the CEO when he was trying to twist my arm to come, I said, you really don't want me. I said, I don't fit. But I have learned a ton in this experience about medicine. Things that I thought I knew being in a university environment for 40 plus years. So common sense about, can I grow in this? And the second point, when students ask me this, I say, you've got to be happy every morning when you get up and excited. And if there's a question about, I'm going to be happy or am I going to be excited, then that's not the step to take. And you need to be proud of the step you took. And if you follow those kinds of processes in thinking your step forward, whether it's one postdoc, two postdocs, whatever it is, whether it's going into industry for a while, whether it's going in this direction or that direction, I think through that carefully, because it's about you. It's not about any of us. It's about what's going to make you happy and excited about your job, and am I going to be able to grow in that job? And those are the key things I think that we need to evaluate each decision that we make. You'll make lots of them. Sometimes you'll make one that isn't quite so good and you need to redirect that quickly, but most of the time you'll make good decisions if you do that. Thank you.
All right, I have a question for Dr. Fordyce, actually. So, working in industry, and you've been sort of on both sides. What are some of the leadership skills that you think were unique to working in industry?
I don't think any of them are unique. I think they are all connected. They come back to Dr. Thompson's comment of, what was your first leadership experience? And they're all relevant to what I've experienced in industry. I think just what was mentioned, it's all pretty particular. My experience in industry has been only 5 years long. It's been only at Gilead. Gilead is a unique company. It's not, every company is not the same. So when we say industry, it's only because of our collective ignorance that we haven't all worked for multiple companies. And I came at a very interesting time where I got to grow into a project and a series of leadership positions. What I learned in my particular transition the last 5 years was about all of the skill sets that's required to develop a drug. In the last 5 years, I was able to collaborate and lead a team from phase 1 through approval and launch of a new HIV drug. And there are so many unsung heroes in that process. I had to organize the celebration dinner when we got this done. I could not pick out of the 8,000 employees who had touched this molecule in critical ways. I eventually got down to 45 people who led large teams, and it was. And I felt very responsible in the way that Dr. Thompson mentioned, that everything is your job. It's the process chemists who got the steps down from 30 steps down to 15 steps so that the cost of production actually made a difference in the developing world for this drug. There were so many little details that were performed. So I guess there's a long way to answer appreciation for all of the different functions that go into developing a drug has been my biggest growth curve going to industry. Process chemistry, manufacturing, every little detail. Last month I was with a sales rep. And when I was a medical student, I had one view of a sales rep, and in January in the snow in St. Louis, going to every HIV clinic to see how our drug was meaningful to those providers and how they were using it with their patients, I had a totally different view of that sales rep. And that was very instructive for me, being a project leader on this team.
So I have another question. Some of us are coming from this program as MD PhDs, we'll have some medical background, and some of us will be purely PhD scientists, all right? So I have two questions for pure PhD, scientists. One is, and even for MD PhDs, as a matter of fact, at some point we'll want to collaborate with other doctors because they have patient populations that we're interested in. How do we get these doctors to buy into our mission, to want to share with us? That's the first part. And then the second part is, how can scientists who don't have the patient exposure directly be leaders in the community? What are some creative ways that can happen?
I'll answer quickly and then I'll hand it off. I think you need to show up. I think face-to-face matters. So if you want to make a collaborator, you have to show up, whether you're learning a skill in a lab and following a tech for a month, or if you're trying to explain your science to a site. You have to show up. And that's not easy to do, and we think we're all connected by technology, and there's just no replacement for it. That's my one piece.
So I think that this is one of the problems of the system and how it is run right now. I mean, how it's been run. There's too much of a separation between the science and the medicine, in my opinion. I mean, I think that it needs to be an integral process. The science is informing the medicine, and as you mentioned, the medical specimens or however whatever is desired needs to be part of the research. We talked about how you need to do human research to improve human health. Learning things in mice might be applicable at the beginning, but you need to translate that to humans. I think there needs to be an engagement, definitely, between science and medicine in order to move both fields, to move translational medicine forward. So how do you entice, if you are not already involved with a physician? Again, it's how you engage with the person, how you make that physician interested in the project. Kind of exactly what Dr. Thompson said before. I mean, you have to engage the physician, make that person interested in what the project is, help them understand why it is critically important that you need samples or whatever else you need, and how to move that forward. I have a very, very excellent collaboration with Dr. Zun. That evolved in that way. I mean, Dr. Zun found something in her lab in the mice, in a cell line situation, and then into mice using interferon on monocytes. And she came to me actually 3 years ago now, and we've been working together to move that to the clinic. And we now have a clinical trial written and ready to go to the ARB and the FDA involving this exact protocol. So she was able to do that successfully, coming to me. I mean, I think that is really, really key to moving science forward.
So I agree entirely with what they said. I want to actually extend the issue. So I think it's face-to-face collaboration. It's really understanding what each other does, forming a true partnership, collaborating on it, not expecting this to be a service, and not appreciating the difficulty of, in a complex clinical environment, collecting that sample or getting that information for you. The other side of it is to understand what's happening right now, which is attacking science at large, which is this issue that you see, and actually most of the generation here that is participating in, is this whole issue of scientific reliability or credibility, right? There are all these reproducibility adventures, there are all these retraction watches. There's concern that we don't do science accurately enough. And I think the one thing we in science haven't held ourselves to get medicine is, I don't think any of that's right, personally. I actually think no one goes into science not wanting to get the right answer, not figuring it out. No one puts out, yes, I understand somebody fakes data, but that's not what we're talking about. 99.9% of science, people are doing it as well as they can. They really believe in what they found. What we haven't held ourselves to as scientists to meet medicine equally is we haven't figured out whether our results are generalizable. And that's going to be important when you get to an outbred population that has complex environments. I bet you 99.9% of every paper, when you get exactly those reagents and do them to the rigor which takes real expertise, that laboratory has done, you'll get the same results. But whether, if I go to another cell line, I'll get exactly the same results, because it's a generalizable principle of biology, is not getting tested before we want to go run right to the clinic, go to an outbred person, take huge clinical resources and say, I want to test it because it's my idea. And we've got to figure out a way to figure out whether scientific discoveries are generalizable before we engage on the medicine when there's real patients with real issues. And I think we've gotten distracted by this reproducibility, and we haven't thought about generalizability. The assay, the test I do all the time, as I said, I grew up in an era where model organisms, or how we did genetics and how we could actually find molecular causes. There are 3 important strains of Saccharomyces cerevisiae. One came from upstate New York, one came from Europe, and one came from South Africa. Almost every lab sticks all their work to one of those 3 strains. They're all able to interbreed. All yeast genetics are done on a single haplotype of one of those 3 strains. There has yet to ever be a genetic screen done in all 3 strains, they're exactly the same species, and get the same answer. Because they're testing a single allele of a population of a species that's been separated by geographies for a large number of years and held separately. We have found incredible things from those screens about how biology can work and how the components can be put together. But if we're going to actually influence medicine, it's got to be generalizable. It's got to not matter which haplotypes you have and all those kinds of things. And we haven't got a mechanism to do that. And I think that's going to be really important. Point number one. The point number two that I want to extend this to is the understanding that doctors don't need more data. And that's the problem with big data right now. Doctors just don't need more data. The rule of the land is every clinical test done on a patient has 95% confidence levels. At the point it exceeds those 95% confidence normals of normal, it has to be reported as abnormal. Patients own their own records. So 5% of normal people on every test are abnormal. And a doctor is both legally and ethically responsible for explaining to the patient why that's abnormal. You give him or her 1,000 tests, and there's not enough time in a day to explain to the patient why they were out of confidence for 133 of those tests. Because that's what it'll be. So you do your whole genome, and they're never going to be able to explain it to somebody. It's got to get down to something simpler. And that's the real problem we have with big data, with molecular testing. We've got to get it down to simpler tests. The example is when I started and John started in medicine, every single chemistry test done on a patient was done on a single machine by a single technician at one time, and you had to order as a physician. In 1977, somebody invented a thing called a serial multiple analyzer, which is just a way to take a tube of blood, run a bunch of fluid in, throw it through 6 different vents, and you can do first 6 tests. We call it an SMA-6. And John and I probably thought it was transformational when we were in our early careers. They got better at it. Engineers get really good at these things. Suddenly they had an SMA-12. Then they had an SMA-18. Then they had, because you can just actually do the channel numbers, they got the 54. The SMA-54 doesn't exist anymore because no doctor could use it. Because you don't need to know 54 tests on everything for every patient because you've had too much time trying to deal with the false positives and the false negatives. That's where we've gotten molecular testing. As we'll talk later, in my laboratory, we now can reproducibly on a human blood sample tell you about 1,000 different intermediate metabolites in your blood. I don't know what you do with that 1,000, but we can send it to you. And so we have a real problem right now in molecular testing in cancer. Right now, the spectrum is right. Right now, all of the pharmaceutical industry, Marshall and his colleagues, are buying a test from Foundation Medicine. It now has just gone over 1,000 genes they assay for. It's actually useful for the kind of big data analysis question. You give this to my doctors who actually take care of cancer patients, and they just throw up their hands and walk away. You have to engage them. So right now, I've got a team of 25 physicians building full annotation on the 34 genes that it makes a difference to know the answer on. And we're marketing that test with Quest because we have to have a commercial partner who can absolutely do the test 100% reliably. Ultimately, there'll be something in between, but it has to be useful to them. The docs, even in an advanced single dedicated cancer center, can get excited about 34 genes that they know why they need to know those because they know some drugs work and don't work on that. You give them the 1,000 Foundation ads, which might be useful for a research basis, they have no idea how to even begin looking at the data. And so you have to actually pair both generalizability and usefulness in when you propose something as part of this collaboration. And that, to me, beyond what I absolutely think that what they said is thing, but if we want to get to really good, we've got to actually address those from the scientific side.
Let me come back a little bit. I think what was being asked about, some of you are going to be PhD and some of you are going to be MD, PhD, and I think one of the luxuries I had in my life, I don't know why I was afforded this luxury, was I was able to grow up in my career on both sides of the campus, both the basic science side and the clinical side. And I think that it helped me understand the differences that we bring to the job each day, the visions that we have of what we want to do, what we want to accomplish. The challenge that I've faced, and I'm sure Craig has faced it numerous times at Penn and his job now, is this kind of, there's basically a fear, I don't know how else to label it, on both sides of the street, And a lot of this, I think, having watched that fear and tried to melt that fear to integrate a team, has been language. And it's kind of language in many parts of our lives, knowing the other individual's language. When I sit down with a patent lawyer, I'm totally lost. I have not a clue what they're writing or what they're talking about. But it's language, isn't it? It's not that that individual's smarter than I am. It's just he's got a different set of language skills. And I've thought over the years that a lot of overcoming those initial hurdles is really getting comfortable with each other. And I used to say, the first meeting's going nowhere, the second meeting's going nowhere, but maybe by the third meeting we'll start to understand each other and what each other is trying to bring to the table, and to build, then begin to build a team. I remember, and maybe Craig does at the University of Michigan, there was an elder gentleman, elder to me as an assistant professor, who headed physiology. And the thing that I was thinking about as we were talking today, as you were talking, is this individual back then, and that was in roughly plus minus 1970, shows you how old I am. He grew up, he never did an experiment with anybody. He washed his own glassware, he was a physiologist, he set up his machines that measured what he was trying to measure himself, conducted that experiment. At the end of the day, put everything away, washed all the glassware and everything else, and that's the way he learned. Today, it is very, very difficult. I would challenge anybody to work alone. You have got today to take a question and build a team to address that question, because none of us have all the language, all the skills, and we have to learn how to build that team. Who's got the expertise in this? Who's got the expertise in that? And that puts other challenges on leadership in order to give credit for everybody that's part of that team. And those are other challenges that we face. But this is teams today to address the complexity of the questions that you want to address. And that means some of you are going to be educated very deeply in science. Some are going to be educated a little bit in science and a lot in medicine, and then your careers will evolve from that point. A lot of my MD, PhD colleagues have gone very, very deep into science and wouldn't know the first thing to do if they were faced with a patient in the hallway. So, I mean, it evolves, but I think it's about building teams today and how to do that effectively. So get rid of the fear part, be willing to work as part of the team.
Thank you. So we're just about that time that it's time to get lunch, but just before we wrap it up, I'd like, if possible, from each panelist, if there's one sentence you could tell yourself, if you were to rewind and you're now in the middle of your PhD or your MD, what would you tell yourself? Besides, like, wash your T-shirt or something.
I'll start because it's just a follow-on to John, but it is actually the thing I think at least I feel like I forgot to mention, which is exactly what he said. The hardest part of medical school is learning the first 2 years of the language of medicine. The hardest part of science is learning the evolving language of science and the techniques we have. And the one thing you learn and get more comfortable with as you get on, and I wish I'd been more comfortable earlier in my career, is when you don't understand what a word means or what somebody is saying, stop right there, and you're not stupid. You didn't not do this. You're here for all the good reasons, and ask, what does it mean? And stop and actually get a common definition, because that's how you get over it. And I completely agree with what John just said. That is one of the biggest boundaries to us working effectively and collaborating, because we each, even in different scientific disciplines between X-ray crystallography and cell biology, we have different nomenclature. You've got to stop people at that point. Actually, probably the best skill I use every day as a leader. I just don't care. If I don't understand, we stop the meeting and they have to explain it to me. Actually, what I've always learned is about a third of the room gets more comfortable after it's explained. I just have to be the one to embarrass myself and say, I don't know, and I don't care. I need to know. And you need to do that as a student, too. And I wished I had learned that much, much earlier in my career. Now I'm fearless about it, but I should have been more fearless much earlier. And I think John reminds us how important that is.
So I think I would echo a little bit of what's been said throughout the day is have an open mind and listen to where things are taking you, and whether that's with respect to what the data are showing you. If they're showing you something that's not what you expect, maybe that's really the right answer. Have an open mind with respect to interpreting the data. Have an open mind with respect to where you're going next. It might not necessarily be a logical path that you planned from the beginning. Maybe certain opportunities are opening up that you didn't necessarily anticipate. So have an open mind with your next steps in your career, and the next steps in your life and your travels. So I would say have an open mind with everything that comes up.
Right, it's hard to keep extending all these good ideas, but you asked sort of a reflective question. I'd say trust yourself. I wish I had trusted myself earlier. You sort of feel like it's not until you've arrived that your idea is valid or that your challenge is important. But you should be challenging early and trust your challenges. So stay open, but don't stay so open that it completely tips you off of where you stand, what you know, and what you think the relevant questions are. So I'd say trust yourself earlier. Don't wait till you've arrived, or don't wait till you got your degree, but trust yourself early.
Gosh, there's so many things that pop into my head that I want to throw at you. I guess that comes with age. I'm sorry for that. I wish I was your age and the excitement ahead of you. The one piece of advice I would tender is don't be in a rush, as Dr. Thompson said, don't be too much in a rush to take on leadership responsibilities. You're all very, very talented. So the biggest problem as your career continues to mature is people are going to pick on you to take on leadership jobs, whether you think that's true or not. I guarantee you, when you start into your 40s, people are going to, look at your accomplishment and are going to throw things at you that are going to be enticing. I can remember deanship opportunities and chair opportunities. And I was lucky enough to have an older mentor who I could turn to every now and then and always reminded me what I was going to lose if I took that on. And I thank that individual who's no longer with us, but I thank him repeatedly because I know my path would have been very, very different. But as Dr. Thompson has said so many times this morning, gosh, what I'm so excited about every day, even at this stage of my career, is I'm surrounded by science, and we're doing exciting and interesting things, and it's fun to do that. So just maybe that's the best advice I can give you today. Thank you.
Thank you so much to all our panelists. This was a fantastic discussion, and thank you to our audience.
So also I want to thank Leanna for chairing that session. Very good. We're not going to lunch yet. We're going to take about a 15-minute bio break, grab some food in the back. I think there's still coffee, and at exactly 11:15 we're going to start a couple videos about what precision medicine is, and a video about a summit that happened at the University of California in San Francisco very recently. Did anyone on the panel actually attend that summit? That's unfortunate. I was hoping to get some insights about what happened. I know, it was like, I figured of 300 important leaders across the country, you guys would have been invited. What's wrong with them? Yeah, exactly. So we're going to have our own mini summit here about, because it's really what they did, is they just got experts from different disciplines, all aspects of healthcare, from scientists to practitioners, and talked about precision medicine and how to make it happen and what it means for health. And so we're going to do the same kind of thing here, just so we begin thinking about what precision medicine is. And we have a series of questions. So we're going to set up 5 tables, so all of you distribute yourself around 5 tables. Our panelists are going to sit at one of the tables. Kathy Zuna is going to sit at a table. So they're going to be the leaders of your table. And you're going to go through these 4 or 5 questions and provide some answers. And then over the lunch hour, we'll have a representative from each table stand up, and we'll have a little mini discussion about what they came up with. So it should be fun. So we'll see you in just a bit.